Okay, so we're continuing on. We have gone through the general overview and general organizational principles of the autonomic nervous system. We're going to continue that today and extend and begin. We're going to be at the threshold of starting to take what I might call the deeper dive into the autonomic nervous system. That is where we're going. But today's talk, this small lecture, is basically going to be to bridge like the general overview kind of discussions to the deep dive by telling you how we're going to go about understanding how the autonomic nervous system, both the parasympathetic and sympathetic nervous system, are mediating all of the effects that they do. So let's just do the quick review, get everybody on the same page and warm our brains up a little bit here. And then I'm going to go through how we're going to begin to take that deeper dive and how I would like to make this seem a little bit less daunting to you. A lot of students start to feel overwhelmed when we get in and start doing that deeper dive. And the purpose of this little segment of the lecture where we're just about to do that is to ease your mind a little bit, because I'm going to give you a rationale about how we're going to go about it, which doesn't seem too bad. All right, so the somatic nervous system, let's just review here. It's voluntary. Its motor neuron starts in the central nervous system, just like those output neurons of the autonomic nervous system do. But the somatic motor neuron, the alpha motor neuron, which is in the ventral horn of that spinal cord, those large alpha motor neurons are going to send their axon all the way out to skeletal muscle targets. Those alpha motor neurons are under voluntary control. Put my arrow there. They will then send an action potential down that axon and release acetylcholine onto skeletal muscle to initiate the process of contraction. So those neurons are cholinergic. Okay. And remember, the autonomic nervous system and somatic motor system are parts of the peripheral nervous system. So as soon as their cell bodies send their axons outside of any of the bony structures that define the central nervous system, we are in the peripheral nervous system. So just like that was the case with the somatic motor system, where the cell body was in the central nervous system in the ventral horn, the sympathetic cell bodies are also in the spinal cord in the intermediolateral column. And remember that, just to compare and contrast, these are involuntary. And these neurons, though, have an outflow to their effector organs, which is not monosynaptic. There's a disynaptic pathway out to effector organs for both the sympathetic and the sympathetic nervous system. So let's talk about that. In the sympathetic nervous system, the two neuron chain is always defined by the first neuron being called the preganglionic fiber or the preganglionic axon. That's because they are talking to a ganglia out in the periphery. Whether you're in the sympathetic or parasympathetic nervous system, each branch has ganglia out in the periphery. So the neuron that speaks to those ganglia is the preganglionic fiber. In the sympathetic nervous system, that preganglionic is releasing acetylcholine, so it's cholinergic. And then the neuron in the ganglia will then be called the postganglionic, and it's going to release epinephrine or norepinephrine onto its targets, okay? Epinephrine or norepinephrine from the sympathetic nervous system, from the postganglionic fiber. Because the autonomic nervous system's sympathetic ganglia are very close, tends to be close to the spinal cord, their preganglionic fibers are short, and because of that, the postganglionic neuron has to send a long fiber out to its effector organ, okay? The neurotransmitters again, preganglionic is releasing acetylcholine, the postganglionic releasing epinephrine or norepinephrine. So these are adrenergic neurons. All right. The parasympathetic nervous system also has the 2 neuron outflow, the preganglionic neuron. Again, one of the anatomical features that we discussed yesterday was the outflow of the sympathetic nervous system. These neurons that were in the intermediolateral column of the spinal cord are coming out of the thoracolumbar regions. These neurons within the central nervous system for the parasympathetic nervous system are coming out of cranial and sacral regions. And the cranial regions means their cell bodies are going to be in cranial nerve nuclei in the brain stem. In any case, the ganglia of the parasympathetic nervous system tend to be far away, so their fibers tend to be long. But just like in the sympathetic nervous system, they are releasing acetylcholine. Because the ganglia of the parasympathetic nervous system are close to their tissues or organs or glands that they're talking to, their postganglionic fibers can be short. And they are also cholinergic. They're releasing acetylcholine. Okay? Where we're going when we take the deep dive into the autonomic nervous system is to ask the question how the sympathetic nervous system and the parasympathetic nervous system achieves its specific effects in each one of these glands, tissues, or organs. I'm going to try to make the deep dive simpler and not so daunting by viewing everything we need to know under a more simple framework. So if this is going to be an effector cell or tissue of any aspect of the autonomic nervous system, parasympathetic or sympathetic, in every case, for every one of these pictures, we're simply going to ask what receptor type is in that tissue, gland, or organ. And obviously, the receptor type has to be a cholinergic receptor or an adrenergic receptor. Okay, those are the only two choices because those are the only transmitters that are released out of the autonomic nervous system. So we're going to ask what cholinergic receptor or what adrenergic receptor are there in those target tissues or organs, and how their signal transduction pathway is leading to the response. That's it. And when we start doing that, we're just going to start seeing common themes happening over and over. Of how, for example, in a smooth muscle cell, so I'll draw a little spindly, smooth muscle cell, how it is that depending on where that smooth muscle cell is, the sympathetic nervous system can lead to increased contractility or decreased contractility depending on where the smooth muscle cell is and what receptor types it's expressing. That'll just give us an idea of like the game we're going to play to understand how we go from the autonomic input to the receptor, to the signal transduction, to the response for every one of these tissues that you see on this complicated slide. And it'll make it seem a little bit more tenable and less daunting to you as we get into that. I also should say, as I get into some of the signal transduction pathway here, I don't want you at this stage worrying about any of the details right now of this signal transduction pathway or the receptors or the receptor types. I just want you to listen and appreciate the idea that different receptors can lead to different signaling pathways that can have different effects in the cell. I'm going to gloss over some things. I'm going to ignore other things in this picture. Just want to give you guys an idea of the framework by which we're going to do the deep dive, where we're going to try to understand the receptors on every one of these organs, tissues, and glands, and how it is that they have their cellular effects. Okay, as promised, I am showing you a smooth muscle cell and a lot of diagrams of signal transduction pathways. And I promised you that we can understand autonomic nervous system specificity and how it controls all of its tissues and organs and glands by having the framework of trying to understand how the receptors are coupling through these signal transduction pathways. So this is a smooth muscle cell. And maybe before I start with the receptors and signal transduction pathway, let me just remind you of the physiology of the smooth muscle contraction. It's a little bit different, well, it's quite a bit different from the skeletal muscle contraction, from the excitation contraction coupling that you have in skeletal muscle, where the actin is sort of buried by the troponin complexes, and the myosins are always sort of ready to go. And once the actin site is kind of exposed through calcium, through the troponin regulatory complex, cross-bridge cycling can begin. This is a little bit different. Here, the rate-limiting step is whether or not your myosin is going to be phosphorylated. When it's phosphorylated, that pretty much can initiate contraction in smooth muscle. So really, let's just introduce that. So the name of the game is going to be regulating phosphorylation of myosin. And whether your autonomic nervous system is going to want to contract or relax a cell is going to either increase or decrease phosphorylation, respectively. And that's going to be it. So let me just show you how it goes. So again, we've got the sympathetic, in this case, the sympathetic nervous system sometimes wants to relax smooth muscle cells, which we're going to see when we do this deeper dive. Some of these smooth muscle cells in these organs want to relax in response to sympathetic stimulation. Might as well tell you that would be, for example, in the airways here. And other smooth muscle cells, like perhaps in the vasculature going out to the skin, you're going to want to shut down those blood vessels. You're going to want to constrict those or contract those smooth muscle cells that regulate those vascular beds. So we have the same tissue type. It's smooth muscle. We have the sympathetic nervous system talking to smooth muscle cells, but depending on what tissue or organ that smooth muscle is in, we can still have differential effects based on the receptors that are in those smooth muscle cells. So I'm just noticing here that we don't even have any receptors. So we've got sympathetic nervous system, and I said they're going to be opposite effects. So I'm going to put in two receptors to show you how we can both increase contractility in some smooth muscle in response to sympathetic nervous system input. Again, let's just put the neurotransmitter there, releasing epi. Or we can have another smooth muscle cell somewhere else releasing epi, still on smooth muscle. So we still have an adrenergic receptor. Okay, but in one case, we're going to get signaling that relaxes. And in another case, we're going to get signaling that contracts. And that's going to depend on the type of adrenergic receptor we have. So let me start up here. So let's see, this one is close to the GQ. So that's going to be an alpha-1 receptor. And this one here by the GS, that would be an example of beta-2. So let me go through an alpha, like signal transduction through an alpha-1 receptor. So that would be here. So sympathetic nervous system, postganglionic, releases its epinephrine. Alpha-1 receptor is going to signal through this pathway. It's going to activate GQ, and not all the steps are shown. I know this diagram looks crazy, but this is actually a simplified diagram. There's a lot of things missing from here. But this GQ will activate an enzyme called phospholipase that will increase IP3. And through a series of steps, IP3 is going to gate calcium flux out of sarcoplasmic reticulum. You're going to increase calcium. That's going to increase calcium calmodulin complexes in the cytosol. And this kinase, this myosin light chain kinase, reacts to the calcium calmodulin by being activated and phosphorylating the myosin. Remember, the phosphorylated myosin makes it so that you want to contract. So this whole pathway I went through is going to constrict or contract that smooth muscle. That might happen when you are in a sympathetic state and you have your blood vessels going out to your skin or periphery all closed off because you're shuttling as much blood as you can to your muscles. Okay. The second example, now, how can we do the opposite? Well, it's just we need a different receptor to signal through a relaxation signaling transduction cascade. So for that, we have our beta-2 receptor. And again, a typical organ or tissue where this would happen, a smooth muscle cell, for example, in the bronchiole, where you are running for your life. Your sympathetic nervous system is on high alert, and you're making a lot of epinephrine, and you're putting it onto beta-2 receptors in your airways. Those airways will open up so that you have maximal ventilation for your poor muscles, which are trying to run away from the tiger. That coupling, that receptor is going to have a completely different signal transduction pathway, which will end up having the opposite effect. In that case, the beta-2 receptor will signal through GS proteins, and that'll stimulate an enzyme that will catalyze the formation of another second messenger. You'll stimulate adenylyl cyclase, you'll make a lot of cyclic AMP, and that is a negative regulator. That is a negative regulator on the myosin light chain kinase. So you cannot, this kinase cannot phosphorylate the myosin. You stay in the relaxed state. So in one case, we had relaxation being dominated, and in the other case, we had contraction, a contraction state in that smooth muscle being dominated. Interestingly enough, both through sympathetic nervous system, depending on what receptor types are dominant in those smooth muscle beds, in those organs and tissues. This is the game we're going to play. We're going to go through every one of these tissues, organs, and glands. We're going to ask what adrenergic and cholinergic receptors do they have, and we'll go through the basic signaling pathways to understand a little bit of the biochemistry of those cellular responses.